Okay, I think we'll get started if that's okay with everybody. And it's okay with our speakers, so that's good. For those who don't know me, I'm Rob Brownstone. I'm the head of the Division of Neurosurgery here at the UCL Queen's Square Institute of Neurology. We started some years ago a lecture series called the Future of Neurosurgery Lecture Series, and it's somewhat irregular, but we like to take the opportunity to have people speak when we can, and we have two speakers today in this lecture series, and I'm really pleased that we have them both, and I'm really looking forward to hearing their lectures, which will be about the future of neurosurgery, from the present to the future, which is very exciting. So our first speaker is David Brandman, who is an Associate Professor at University of California, Davis, where he is also Director of the Functional Neurosurgery Fellowship and a co-director of the Neuroprosthetics Lab. You make sure I get everything right, David. Okay. He is also affiliated there with the Center for Neuroengineering and Medicine and the Center for Mind and Brain, if there's a difference between those two, and is part of the graduate group in computer science. So David, I've known for some years. It's always a pleasure when somebody who was your trainee does so incredibly well. And I can't take any credit for David's training at all, other than that we crossed paths and he was a trainee in our neurosurgery program in Canada at Dalhousie University. Prior to that, he did a Bachelor of Science in Biophysics at the University of British Columbia, then went to the University of Calgary where he did his medical degree and joined us on the East Coast. So that's coast to coast in Canada, for those of you who don't know, which is farther than it is from the East Coast to London. Anyway, he joined us, I think, in about 2010, maybe, to join our neurosurgery residency program. And he was a bit of an oddball in the sense that he came from this biophysics background. And my memory of David when he joined our program was that he had this vision of combining robots and people into one sort of organism. and anyway, we talked a lot over his first few years there, and until he realized that he needed to spread his wings a little bit and went to Brown University, where he did his PhD with, I think primarily with Lee Hochberg. Is that right? With Lee. He came back after I was gone, I guess, to Halifax, finished his neurosurgery residency, and then went down to Bob Gross's empire at Emory University for a functional neurosurgery fellowship, and he completed it there and was shortly afterwards recruited to University of California, Davis, where he currently is. David is quite the guy. We are going to hear a fantastic lecture. I have no doubt about it. He publishes well. You may have heard of some of the journals he publishes in, like the New England Journal of Medicine and Nature and Cell, and also probably in a few journals that you haven't heard of, but that's okay too. He's very involved in lots of clinical trials. He is a popular speaker, has a number of visiting speakerships on his CV. He's well funded from a grant point of view, currently holding an ALS Association Assistive Technology grant, and is co-PI on a Google Research Scholar Award as well. He's won a number of prizes over the years, including being included in the Time Magazine 100 Most Influential People in Health in 2026. So it's a real pleasure to have David here to tell us I can't even read his title here. Intracortical brain-computer interfaces to restore communication in people with paralysis. I'm really looking forward to this. Thanks for the introduction.
Thanks so much for that, Dr. Brownstone. As an intern in neurosurgery at Dalhousie University, you were super intimidating. So it's cool to be in this position now and however many years later and be speaking where you're now chair. So thanks so much for the invitation to speak. I'm going to talk to you a little bit about brain-computer interfaces today. I am currently an associate professor at UC Davis in Sacramento, so it's about an hour and a half driving distance outside of San Francisco, just from a geography perspective. It's a 10-hour flight to get from San Francisco to Heathrow as part of this. So it's pretty far away, and going from Vancouver to Halifax is probably the distance of flying going from London to Moscow, or perhaps even further. So Canada's a really big country. So this is my second time to the UK. First time was here when I was backpacking before starting residency. Now I get to come here as an attending to talk about my research. As we get started, a few things to know about. Number one, I am an ad hoc consultant for a surgical device company that builds robots, and I am going to be the proposed study principal investigator for a clinical trial put on by Paradromics Incorporated. I'm going to talk a little bit more about that as we go on. I'm going to start with a video. And in fact, the way I'm going to structure this presentation is showing you the end, the culmination of the research, and then I'm going to go through and talk about the stepping stones it took to be able to make this happen.
Not being able to communicate is so frustrating and demoralizing. It is like you are trapped.
Communication is so important to our everyday lives. Something as simple as, I have an itch, or I'm thirsty, or I love you. Imagine not being able to say those things to the people that you care about most in your life. A brain-computer interface is a device that records brain signals from people that are paralyzed, and then translates those brain signals to allow people to communicate. So this research is happening as part of a clinical trial called BrainGate 2. The idea is people living with conditions like spinal cord injury, brainstem stroke, or ALS undergo surgical implantation of electrodes into their motor cortex. And thereafter, they work with a scientific team to develop next-generation technologies to help people living with paralysis. I recently implanted a 45-year-old man with ALS who really can't be understood when he tries to speak very easily. And by using a BCI, we can translate his desire to speak and have words appear on the screen.
In essence, what we're doing is we're bypassing the injury. We're recording from the source, from this part of the brain that's trying to send these commands to the muscles. And we're translating those patterns of brain activity into the phonemes, so that's like a syllable, the unit of speech, and then the words that they're trying to say.
Not only can we accurately decode the words that he's reading prompted off of a screen, but we can also accurately predict what he's trying to say in free-form conversation.
A lot of the demonstrations of BCI technology over the past few decades took weeks or months to get working. The first time he tried to use the system, it just worked.
Okay, ready?
That first time that he tried to speak and that word appeared on the screen, he started crying, I started crying, his family started crying. Basically everyone, we had to pause. We stopped the rest of that block of experiment trials. Everyone composed themselves. They were like, all right, let's do it again. And it kept working. So yeah, it was really special. It was a special moment.
At this point, we can decode what he's trying to say about 99% of the time, which is better than a lot of commercially available smartphone applications that try to interpret your voice, which hover around 95%. This is by far the most accurate decoding ever reported, and it's also a demonstration that this just works.
So it's not that we're reading people's minds, we're not detecting their inner thought or their inner monologue or even their mind's voice. We're really detecting their attempt to move their muscles and to talk.
People who do not have the ability to communicate well or quickly are often isolated and lonely. Having something like this will give them hope and opportunity to be more of themselves.
We've actually used artificial intelligence to mimic his own voice.
The easiest way to protect ourselves from mercury pollution is to limit our consumption.
By using video recordings that were collected prior to him having ALS, we were able to digitally reconstruct his voice. So now when he tries to speak, not only do words appear on the screen, but it sounds like him.
It feels a lot like me. It makes people cry who have not heard me in a while.
I think we're at a point where it does work well enough that it really lets people just be a part of society and to talk.
I am able to still work full-time for climate action, and I am able to do that well and more efficiently with this device.
Honestly, the reason this is so transformative is because it provides hope for people living with these kinds of diseases, that as they face their disease, that they know that they will hopefully have the ability to retain communication.
I hope that we are at a time when everyone who is like me have the same opportunity as I do to have a device like this that will help them communicate. Let's all make that happen, okay?
So we're going to spend the rest of the presentation unpacking what you just saw and talking a little bit how the technology works and where I think the technology is going. So a brain-computer interface is a device that records brain signals from people that are paralyzed and then translates those brain signals to allow people to control objects in their environment. So fundamentally, a BCI has three parts to it. There's a sensor, a decoder, and an effector. In other words, there's a source of brain signals, there is what you're trying to control in the world, and then there's a math that connects the two. And the video you just saw is using a technology to record brain signals called the Utah array. So what's the Utah array? It's a device that's about 3 millimeters in size, and the 3 millimeters by 3 millimeter device has about 64 shanks. Each shank is about 50 microns in diameter that's designed to record from the cortex. The idea being that as electrical activity, as the motor cortex is active, the tip of those tines of those electrodes is recording action potentials, and information is then taken from those microelectrode recordings through a golden wire to a pedestal that sits percutaneously. So these devices are implanted as part of an ongoing clinical trial called BrainGate. BrainGate has been going on for the last 21 years now and is an early feasibility study looking at the safety profile of chronically implanted electrodes in people with paralysis. So it includes folks with spinal cord injury, brainstem stroke, ALS, neuromuscular diseases in general, cerebral palsy, and amputations. And as part of the clinical trial, patients undergo implantation of these microelectrode arrays into motor-related regions of the brain. And the first 17 years of the clinical trial are really based on studying point-and-click communication. Motivated through decades of research in non-human primates, trying to understand how the brain controls motion, the first years of the clinical trial really focused on robotic arm control, cursor control for point-and-click communication, and so forth. And in the last few years, the focus has really shifted onto speech. The idea being that we want to be able to empower people to communicate for those that live with paralysis. Now, I want to be very clear, this technology is not focusing right now on people with aphasia. It's focusing on people with dysarthria. It's not focusing on people who have cognitive dysfunction with language, but the mechanics of taking what I want to say and being able to say that are not working well. And so our first report of this technology came out in the New England Journal of Medicine, where we talked about Casey. So Casey was the man you just met. He was at the time of implantation, a 43-year-old man living with ALS. For those of you who know the ALS-FRS scale, he had an ALS-FRS score of 22. So he's effectively tetraplegic and has very severe dysarthria. So there were two people in the world that could really understood him when he speaks, and I could understand about 10% roughly of what he said when he spoke. And so as part of the BrainGate clinical trial, I implanted 4 microelectrode arrays into the ventral precentral gyrus. And for those of you with a neuroanatomy background in the audience, I went into ventral 6V, dorsal 6V, area 55 for the implantation. And essentially what's happening is that we are taking his intention to speak as reflected by neural activity and then translating his neural activity into words. So every 80 milliseconds, we record from about 250 neurons, and we take the 80 milliseconds worth of neural activity, of action potential activity, and translate that to one of 39 different English phonemes. So what's a phoneme? A phoneme is the building block of language. So if I say the word hello, I can break up the word hello into ha, and o. So this is not a mind reading device, right? We're not reading his deepest, darkest secrets, right? All we're doing is taking his intention to speak and then translating the neural activity associated with that speech into phonemes, and then using artificial intelligence to take those phonemes into words, words into sentences, and sentences into paragraphs. Right? And so this work was led by my postdoc, Nick Card. By the way, he's on the market, so if you're interested in hiring a world-class scientist, please let me know. I can introduce you. So this is one of the main figures from that paper, the idea being that we did the surgery, and then 3.5 weeks later, we showed up to his house, and we turned it on. What happened is we arrived, we donned the system, we turned on the computers, and we just had an instruction set where we asked him to read words presented on a screen. We started with a limited vocabulary, and that was deliberate because that was the previous state of the art reported through different kinds of technologies, through Eddie Chang's work. And if you may have, I think Eddie Chang was here recently talking about his work as well. So based on the previous standard of state of the art, we started with a limited vocabulary. We got about half an hour's worth of training data, and then we flipped the switch. That is, we went from analyzing or reading what he was trying to say to trying to decode the neural intentions, trying to decode the neural signals and turn that into words. And the very first time we turned it on, it just worked. It worked with 99% accuracy from a limited vocabulary of 50 words after half an hour of training data. So that was pretty cool, and that was reflected in the video that you just saw. And then 2 days later, we returned to his house, and we turned it on again. And this time it worked even better. It went from 99% to 100%, and we got excited. And so we turned off the training wheels, took off the training wheels, so to speak, and went from 50 words to 125,000 words. Now, I'm a native English speaker, and I went to college, but I'm not. I don't have the most expansive vocabulary. My vocabulary is probably in the 50 to 60,000 word range. So I recognize many people from here come from very prestigious colleges in the UK, and you probably have vocabularies in the 100,000 range, but very few people know 125,000 words. So this is more words you could possibly want to use. We turned it on, and it worked. It was about 91% accurate. And as we used it more and more, it got better and better. And there's a few reasons for this. One is our maths, not our math, got better as we used it, in the sense that we fixed bugs and we updated our decoding algorithms. We did things with regards to addressing some of the signal characteristics and so forth. The second is we got more data. These algorithms are very data hungry, and the more data you provide, often the better improvement that is. And third, we just fix bugs in our code, right? We are a research laboratory. We discovered that we had bugs, and we squashed them, and the decoding performance continued to improve with that. And so its initial paper, we report performance up to most of a year, and demonstrating that the performance stays pretty consistent. So how fast can he talk using the system? Well, as I'm speaking to you right now, I'm speaking somewhere in the 100 to 150 words per minute, depending on how excited I am in conveying this information. So he's speaking at about 30 to 40 words per minute. So this is not a natural pace, right? This is substantially slower. But it's way faster than his other means of communication. So when he tries to speak through his wife, in other words, his wife is translating his severe dysarthria and trying to get his message across, he communicates about 6 correct words per minute. And when he communicates with his favorite assistive communication device, in this case, a gyroscopic head mouse, again, communicating about 6 words per minute. So we're talking about a jump from 6 words a minute to about 30 to 40. And what we found with time is that it's gotten better and better. So we have this protocol set up in the BrainGate clinical trial where people undergo surgical implantations and they work with our scientific teams a handful of times a week. But the system started to work, and it started to work really well. And so we started coming to his house 3, 4, sometimes 5 times a week to allow him to speak for 8, 10, 12 hours a day. And at this point, we decided to petition the Food and Drug Association to change the clinical trial protocol so that his care partners could actually don and doff the system without us having to be present. And there he was using the system from a few dozen hours a week to using this for 50 to 80 hours a week. Right, so this becomes his primary mode of communication. This is the way he prefers to communicate. This is what empowers him to have communication, to have enough communication for him to go back to work full time. Now again, this is a man who's tetraplegic with severe dysarthria with end-stage ALS, and he's working full time using this BCI. He's got a 5-year-old daughter, and he uses this BCI as his primary mode of communication to speak with his daughter. This has been life-changing for him in terms of being able to restore autonomy and being empowered to say whatever he wants to say whenever he wants to say it, as long as he's using the system. And what's interesting, and something I never considered as a clinician, is that if people have trouble understanding you and you have to go through an intermediary, that puts a lot of pressure on the intermediary. It puts a lot of pressure on your care partner for a few reasons. One, every minute that they're by your side speaking on your behalf is a minute they can't take care of the kids and they can't work or they can't take care of their home or rest, right? 2, it's put a lot of pressure on that care partner because they have to be at their side making sure that they are translating appropriately and making sure they're getting it right. And actually, that's really stressful for people to know that you are the only way that this person could be heard in the world. And so one of the really amazing side benefits of this kind of BCI technology is that it's empowered his wife to be able to have a lot more free time and to look after the child and the household and details and so forth, right? So it's been really cool to see. So not only that, he's used the system to Zoom and so forth, and as we mentioned in the video, we've actually replicated the sound of his voice. We deep-faked his voice, right? And so the idea being is that when the computer speaks, it sounds like him, which he's found a phenomenal attribute of the system. Okay, so not only do we do this highly structured way of communicating, but we actually built him a custom user interface that he controls with an eye tracker. He can also control with intended movement of his arm, in other words, cursor control, and we have a paper on that in the Journal of Neuroengineering showing that you can actually decode cursor control from the speech motor cortex. Be that as it may, we looked at its performance just under 2 years, and it turns out we get it mostly right most of the time, right? It's not perfect. Like it's not 100% accurate. It's somewhere in the 97 to 99% accurate when it's quantified in a very structured way. But gosh, when you are speaking in paragraphs like this man does, if you have a 50 to 75 word paragraph you're speaking, you have a handful of words that are decoded incorrectly. We actually built him a user interface that he could select which word was incorrect, and using language models provide predictions of what he probably meant to say, the same way that your phone provides predictions in terms of what you probably meant to say if you type something incorrectly into your iPhone, right? Sometimes we get it wrong, right? And there's reasons why we get it wrong. Perhaps our model doesn't have enough context. Perhaps the sentence was too short. Perhaps there was transient noise in our neural signal and so forth, right? And so we quantified this over 180,000 sentences, 180,000 real-life sentences, and we get it right all the time, 100% right about half the time, and then somewhat right most of the time, and sometimes we just don't get it right at all. And we blame this on our lack of understanding of the neural signals, and we blame this big on signal non-stationarities, and a myriad other reasons as to what's actually happening under the hood. So everything I've shown you up to now has been empowering a man to communicate through text. But of course, we don't communicate through text messages. We communicate, well, let's try that again. Text messaging is a really good way to be misunderstood when you communicate, right? But as I'm speaking to you now, I'm using words. I'm not speaking to you with speech bubbles, right? And so the next research I want to highlight to you by our then postdoc, now research scientist in our group, Maitri Wegrekar, is answering the question, can we synthesize his voice? Can we go from the intention to speak and actually generate his speech in real time? And in fact, in the video I'm about to show you, that's exactly what we did. And then this is the hard scientific problem, and I'm happy to answer why. But the long and the short of it is, if you have a man who's paralyzed and you're trying to translate his voice as he's trying to speak, you don't know what ground truth is. And it turns out you have to be very systematic about trying to recreate a ground truth from the neural signals and make some pretty principled assumptions about how the data is structured to be able to proceed with this. Now, in the video I'm about to show you, I would encourage you to watch the first video. It's going to be several trials. We're going to watch the first trial and I encourage you to close your eyes so you're not biased based on what's on the screen. And check to see how well you can understand what's actually being said. Okay, let's get started.
I will get there as soon as I can.
Okay, let's close our eyes. That was, I will get there as soon as I can.
Never want to know that there.
I never want to go back there.
I would love to meet your family.
I would love to meet your family. Okay, and this goes on. The point is, that's somewhat intelligible, but that is real-time synthesis with a 10 millisecond delay of a man trying to speak who's otherwise paralyzed. Right, so that is arguably the most intelligible real-time synthesis ever described. we're recording from 250 neurons in the cortex. And of course, there are way more than that we're not recording from. We are dramatically undersampling this system. So everything I've talked to you about so far has been from an ongoing early feasibility study. So in American parlance, what that means is that we have a device, we've asked the FDA for approval to study this device, and we're studying the safety profile of it. And the side effect of being part of this particular clinical trial is you get to study how the brain works and then capitalize on the insights to be able to build prosthetics. But this is not a medical device, right? So I implant devices for a living. Most of my practice in Sacramento focuses on neuromodulation. And when I have neuromodulation case, I have a fully implanted wireless device, right, that works 24 hours a day, 7 days a week. When it does need support, there is a 1-800 number you can call for this kind of support to make it happen. And we're not there yet. I'm not yet at a point where I can prescribe one of these devices, but I think we're getting there. I think, and this is my opinion, I think we're about 5 years away from getting to a point where we can talk to a patient and say, I'm really sorry, Miss Smith, you've been diagnosed with ALS. I don't have a cure for you, but I have a way for you to maintain and retain your humanity through this process. So you can speak on your own behalf, so you can tell your family you love them, and you could articulate to people when you're in pain or when you're uncomfortable. And I think realistically we're on the horizon of 5 years away from being able to do that. And the reason we're about 5 years away is because we're getting to a point where we're understanding more about how to translate these brain signals. We're getting better at neural decoding. And there are now multiple companies in this space that are developing fully implanted wireless systems. Okay, now I have to, again, circle back to my conflicts. I'm the proposed study principal investigator for the clinical trial from Paradromics. So they're developing a fully implanted wireless systems targeting speech restoration in people with paralysis. Right? There's 3 clinical trial sites. There's us at University of California, Davis. There's University of Michigan and Mass General Hospital right now. And I'm hoping in Q1 or Q2 it's almost Q2. So in Q2 of this year, we'll be launching the clinical trial. So I think any day now, we'll be ready to start recruiting participants for this clinical trial. There's other companies in this. So Synchron, for example, has talked how they've closed up 2 early feasibility studies looking at their fully implanted wireless device. This device isn't designed to record from individual neurons, but rather records through the superior sagittal sinus and gets collective neural activity for motor-related cortex. Precision Neurotechnology has a micro-ECOG system they're developing and wants to move into BCI applications within the next 2 to 3 years, according to their scientific team. And of course, Neuralink, everyone here at UCL is familiar with, so I won't go into that. So the one thing I want to highlight in my presentation is that this technology is transformational for people, right? This is not something we can prescribe. This is a N of 1 demonstration of the power of this technology, but this N of 1 has set a new gold standard in terms of what it means for BCIs to be able to store communication. And so the last video I want to play for you before I take questions is Casey, in his own words, speaking about how this technology has been transformational for him. So I went to the BCI Society meeting in Banff, Alberta, Canada. It's about 45 minutes from where I grew up in Calgary. And prior to that presentation, I was asked by some of the organizers if I could collect some videos of BCI participants of what it means for them to have a BCI and how important it is to them. And that's all I did. I asked Casey, hey, what does this BCI mean to you, and what would you like to share with other BCI researchers?
It is also important for you to think about people like me, who cannot move almost any part of their body, even if it is impossible to truly understand what we are going through on a day-to-day basis. This decoder and technology is enabling me to do so very much and stay tethered to my life as a human being. It is sad to say that people in my condition are incredibly lonely because we are often shut out from society, because we do not move at the same speed as the case of everyone, and do not talk or speak at the same speed as well. Imagine that you have technology to be able to communicate, but it was only about 1/10 the speed of what a normal human would call. And then you will have this technology, which is enabling me to keep up with the flow of conversation if I am in polite company and people slow down just a little bit. This is enabling me to talk with my family and my friends and have a productive relationship. With my daughter, who is only 6 years old and does not remember my voice from before, this disease took it away. This device is approximately 5 to 7 times as fast as any commercial products on the market today, and enables me to keep my job as a person who, like you, is doing important work on the issue of climate change. This also enables me to have employment when I have a disease that is so incredibly expensive to care for, and I am not bankrupting my family. It is so emotional to talk about this because this technology is everything to me. I want you to get working as hard as you can, but get this technology into people as soon as humanly possible. It will save people's lives and change people's lives. Your hard work is going somewhere tangible. And I encourage you to talk to the team that is at the conference from the University of California, Davis, to get their reflections about how their clinical work is making a human impact on someone like me.
So I want to say it takes a village to make this happen. So we are one of 4 clinical trial sites. We're also affiliated with the Stanford University, Mass General Hospital, and Emory University. And I run a laboratory together with a scientist. So I'm not a technician working for him, nor is he a scientist I've hired. We're actually co-PIs, and our contract at UC Davis is actually specifically states that we are co-PIs running a laboratory together. So I write grants and supervise students, and we are co-senior author in everything that comes out of our laboratory. That's Sergey Stavisky. We have right now 3 postdocs, a research scientist, 4 PhD students, and some lab members that kind of help support this infrastructure. So thanks very much for your attention. Happy to take questions.
That was a terrific talk, David. Thank you. I'm sure there are lots of questions.
Do you foresee in some sort of at some stage where due to neurodegradation, where this kind of device, where this patient can't use this kind of device anymore?
Yeah.
Or how long do you think he can use it?
So I'm going to repeat the question. So will this device ever stop working because of longevity issues, some kind of degradation of either the signal or the device itself? So up until last month, I had to be quiet about this, but actually we have a paper that's coming out of bioRxiv talking about longevity of his particular system. And we are getting really high quality signals that we can continue to use over 2.5 years after implantation. Okay. So if you ask the question about Utah arrays in general, yes, what you find in the non-human primate world and in some human instances as part of our clinical trial and others is that you are unable to resolve action potentials from individual neurons on the order of months to years after implantation. But that's a different question than asking, can you get a viable signal to control a BCI? And the answer is yes, you can, even if you can't resolve action potentials. And I'll give you 3 examples. So Hochberg 2012, which was a demonstration of a woman with a brainstem stroke who effectively had locked-in syndrome, using a robotic limb to give herself a cup of coffee, that was collected 5 years after implantation. And they couldn't resolve action potentials anymore, but there was sufficiently rich local field potential there to be able to solve that problem. Right? Some of the highest cursor control performance reported prior to Neuralink, right? But although Neuralink hasn't reported it officially in peer-reviewed literature, but what they've reported on X, formerly Twitter, as part of this, was looking at implants that are years after surgical implantation, right? So it's a different question to answer, does it work versus can you see neurons? And it seems like in the interim analysis, the BrainGate clinical trial, the answer to does it work is yes, and the answer to do you see neurons is it depends. Does that answer your question?
Yeah. Going on from that, in your experience of this, which is able to pick up neurons, and other technology that doesn't pick up neurons picks up LFP signals, for example,
yep.
Do you think it matters in terms of performance, longevity, what you are measuring in order to decode?
So there's 2 questions there, if I may. So the first question is, does it matter the source of your signal with regards to action potentials versus local field potentials versus aggregate signals like electrocorticography or EEG? And the second question is, if you have all these technologies, then why does it matter? Like, how do you pick the right technology, right? So there's really 2 questions in there. And I'll answer the second one first, because it's easier. And that is, I foresee that much in the same way that my epilepsy practice, I can offer deep brain stimulation, responsive neurostimulation, vagal nerve stimulation, laser ablation or resections, or disconnective surgeries, right? I foresee that within a few years, not 5 years, but beyond 5 years, there's going to be BCI centers of excellence, much like you have epilepsy centers of excellence. They'll be able to offer a Synchron device, a Paradromics device, a Neuralink device, and whatever other companies on the market right now. And so you're going to tailor the device for the patient. This patient's goal is to call for help at 2 o'clock in the morning. Okay, you don't need a BCI implant. You don't need surgery for that, right? You need an EEG cap. This other patient's goal is to go back to work full time. Okay, you probably need neurosurgery for that, right? And there's going to be various kinds of pros and cons of getting an open cranial surgery versus an endovascular surgery and so forth, right? So I foresee these centers developing where you have local expertise of surgeons working with neurologists, working with physiatrists, speech-language pathologists, and so forth, that are going to be able to prescribe the right device for the right patient. Okay? Now, the answer to the first question is, at the end of the day, I don't care, right? My job is to restore communication, right? And the fact that it has the form factor of a DBS versus a VNS system, a deep brain stimulator is a vagal nerve stimulator, I could care less. I want what's most efficacious for my patients, right? And it really depends on your risk-benefit for that specific patient you're working with. So right now, in my own hands, I have the most experience with Utah arrays. And so if you were to ask me, how would I go about building a BCI to restore communication, I would use a Utah array. I think if you were to ask other surgeons with more experience with electrocorticography, they would say electrocorticography is the answer. But I think the data, at least for Casey, speaks for itself, that this is by far the most effective communication neuroprosthesis ever developed. Is that fair? Yeah.
So you said, of course, your prediction is incredibly accurate, but you said sometimes your prediction is not perfect, possibly related to context. So my question would be, at the moment you are recording only from the placentral gyrus, are you considering recording outside the placentral gyrus to give more context to your speech?
Yeah.
If you are recording from the placentral gyrus and you have more information, not only motoric or speech, but on other aspects that could potentially read the mind, what do you think are the implications of this type of processing? Because there are other people, Ariel Goldstein, for example, who are doing this, and of course it has also implications.
Yeah. So there's a few questions to answer that. I want to make sure I get the questions right. One is, what are the potential ethical implications of reading from parts of the brain outside of the motor cortex, and how do you plan to solve all these problems? And the first part of the question is, where ought you go to the brain to solve this particular problem for my patient? Does that encapsulate it, part of that? So I think much in the same way that you think about deep brain stimulation as having an implant that solves a particular problem for a particular patient, I foresee BCI technology is going to have a particular implant destination for a particular indication. So this is probably the right answer for people with dysarthria and arthria. It's probably not the right answer for people with a left MCA stroke. And so the next question to ask is, where ought you go for a left MCA stroke? And the answer is, I don't know, but I just submitted a grant to the NIH to figure that out. Right? So the people are asking those questions of, just taking a step back, what's my job as an academician? Right? My job as an academician in this field, now that there's companies that are building devices to do these kinds of things, is where do I go in the brain and who do I help? Right? What are the right indications and where are the right cortical or subcortical targets that help me get the information I need to bring into my decoder to solve my effector problem? And I very much think that there might be the role for motor speech areas in people with stroke, maybe the contralateral hemisphere, maybe perilesional tissue. Or maybe we have to change our approach to the coding altogether and look for semantic type information. Right? And the reality is we don't understand how stroke is encoded at a single neuron basis. I was about to say no one, but there's actually a German group that's doing this, but almost no one in the world knows how this kind of stuff is working, right? And so our job is to plumb the depths of the brain as neurosurgeons to be able to solve these hard problems and then to be able to demonstrate the feasibility so that these companies come in and run the proper clinical trials that show it's safe and that it works. Okay. So then the second part of the question you asked was about improving predictions, right? And, I'm not sure if there's an open beer session afterwards, but I'd be happy to answer that in more detail in terms of the ethical implications of the technology. I think it is the right time to start asking for these kinds of questions, but I want to steer us to the here and the now. I think that in our field, there's a lot of transhumanists, people that have very grandiose ideas about man merging with machine, and regardless of how you feel about that, the point is there's a man who's paralyzed who wants to talk, and I want to help that guy, right? And so I think this is where the technology, really, the rubber hits the road. We're actually able to effectuate change or demonstration of the feasibility of this technology effectuating change, and we have to run with that as surgeons. Is that fair?
Thank you so much for your talk. It was really interesting. My question is, in your experience what actually works for maintaining performance as arousal and fatigue just different over time?
So,
What doesn't work?
yeah, great. So the question is, look, people get tired. Does your system still work? Right? And the answer is not as well. Yeah. So it turns out you have to be awake to be able to speak, and you have to be paying attention for the system to work. So arousal and attention are important for this. And why is this especially important for ALS? Because people with end-stage ALS are tired all the time, right? Just being upright in a wheelchair is incredibly fatiguing for people, right? I have another patient we have just reported on. Yeah, so it's a paper that's about to come out, so I can talk about it, about a man who has a locked-in syndrome, right? So he's further along in ALS, and when I met him, all he had was extraocular movements and forehead elevation, and now he really does not have forehead elevation. He's got limited eye movements, and he's fatigued just by trying to pay attention throughout the day. So yeah, that is absolutely an important limitation of this kind of technology. Just like a car is really useful to get you from London to Edinburgh as long as you're awake to drive the car, right? So an assistive communication device is going to be helpful for you only if you're awake enough to be able to use it.
Sorry, and the other half of the question was, have you seen any solutions to deal with that drift? That was the other part of it. Have you troubleshooted solutions?
Yeah, so the second question is, how do you fix this, right? So if you have to be paying attention to be able to use a system, how do you address someone's attention? And we haven't specifically investigated that because we're focused on the practical problem of, if you're awake, you're probably not going to want to speak if you're sleeping.
Very impressive talk. I'm a pediatric neurologist. I just wanted to ask you two things. One is, does anyone has tried any of these devices in people with disorder of consciousness, in different minimal conscious state, or to see if actually what in some of these cases people are saying that they are seeing on fMRIs, if they can actually express more than what they do. And the second question is a bit more theoretical, philosophical. I don't know if you envisage that actually these devices in the future can be used for developmental disorders, so that people can get taught actually how to interact with the world when actually they don't have the ability of this.
Yeah, so the first question is about, have we thought about using this in minimally conscious states? And the second question is about developmental disorders. Have we thought about doing that? So stay tuned for the first question. I can't talk about it now, but I hope in the next few years I'll be able to talk about that. And then I will say in terms of your second question, so right now we have not looked specifically at developmental disorders, but you could definitely imagine it having a role in that. Certainly the clinical trial enables people with cerebral palsy to join, right? So right now the minimum age for the clinical trial is 18. You could imagine a world where you introduce this kind of a technology to a 12-year-old who's with it, who's in there, but is simply their body doesn't function the way you expect them to, and chances are without some kind of intervention, their neurocognitive development won't continue, especially with normal language, right? Could we somehow use this as a rehabilitative technique in that kind of patient population to do good? No one's done that in kids. I know this because I sent an email to Adam Curtin, a Canadian gentleman who's at the University of Calgary Pediatric Hospital there, and he's probably one of the leaders in the field of pediatric BCI programs. So people looked at EEG, electroencephalography, for that, and it turns out, yes, kids can pick up how to do this. You can have spastic quads that are nonverbal that can pick up how to use EEGs and play video games. Kids love playing video games, right? And they can use it as a communication device, a limited communication device as well. As far as I know, no one's done Utah array implantation or some kind of neurosurgical procedure on that patient population, but I think there's an appetite to do that. I'm not aware of anything else outside of other neurodevelopmental conditions. So I'm not aware of anyone doing this for Parkinson's disease, for example.
Yeah, I think back to the question about arousal and attention. How much of this is like brain activity being used to train a decoder, or a decoder training brain activity to do something?
Yeah.
Because if you're like not maybe paying attention, like it's actually that you're not using the decoder the way that you want to intend to.
Mm-hmm.
So it doesn't actually matter if you're specifically in the specific part of the motor cortex that is involved in speech, but more that you're training and controlling a decoder to move in a specific way. You mentioned that you could also move a cursor using that same implant, right? So maybe that is more training, and it could be that actually whatever is the most convenient part of the brain is going to sort of map onto. should be where the decoder should be placed?
Okay, so there's actually 5 questions there. I'm sorry. I'm going to have trouble keeping track of all 5 questions you've asked. I'm going to start by the neuroscientific question you asked first, which is, okay, you have the brain's doing something, and we're training the maths to match the brain versus can the brain learn how to match the maths, right? And this is an ongoing scientific question. This is not something that's really studied in people. It's studied in non-human primates. The answer is everything we've done here in 99.9% of the human literature is about adapting the maths to the person, right? So when we got Casey to speak for the first time, we spent our algorithms to learn what those brain signals were, updated the parameters and the hyperparameters of our neural network to be able to translate his brain signals into phonemes. We didn't come up with an arbitrary setup of parameters for the neural network and then spend hundreds of thousands of hours of him speaking to learn how he ought to be able to speak to be able to communicate. My PhD work was actually on cursor control. So it's how do we get people to communicate as quickly as possible through point-and-click communication? And the answer is it takes seconds. You can have someone, a naive BCI user who's never used a system before, you give them the motor imagery instructions to control a joystick, and they can start acquiring targets within 37 seconds of using the system for the first time. That's because you're adapting the maths to the person. Now, you can ask the question of monkeys, right? Because monkeys will work for hours, day after day after day. You can take a linear decoding, something that just takes the brain signals and has a linear mapping to cursor control, and ask how long does it take the monkey to learn how to do this? And the answer is it takes days to weeks for the monkey to figure out how to make some kind of motor action to map that reliably to cursor control. A monkey can learn it, and the caveat is that it depends on how the parameters are set. So a monkey cannot learn an arbitrary mapping. There's a subset of mappings that it can work with, and some that it's never going to be able to learn. And it turns out you can start rotating what those parameters are, rotating in the sense of moving those parameters according to some two-dimensional space, and there's going to be a subset of parameters it can learn quickly and others that it can never learn. So the brain is wired to be able to do these kinds of motor activities in very precise ways, and sometimes the parameters happen to work out in such a way the brain can learn it, and sometimes it doesn't. Now, that was question number 1. And so I'm happy to go through the other 4 you had, but is that sufficient to kind of... Okay, thanks.
If there are no other questions, gone. Thank you, David. That was a stimulating talk.
Appreciate it.
Recording talk.
Thanks.
Thank you very much. That's right. Okay. All right. That was terrific. That was really an enlightening talk, so thank you very much. So our next speaker, who's coming up to set up his computer, is Kai Miller, who is from, was there, no? I thought I heard something.
I thought I heard something too.
Kai's from the Mayo Clinic in Rochester, and was here. I met him a few years ago. He was here giving a talk in the Wolfson Lecture Theater. I can't remember what the occasion was, but that is, I think, the only time that I've met you, Kai. He is a Californian, trained initially at UCSD, went to University of Washington, where he did a PhD in physics, and then an MD, and if that wasn't enough, another PhD in neuroscience. His MD, PhD was part of the MSTP, the Medical Science Training Program, something that America started in about the 70s. That is absolutely fantastic and a great way to produce star clinician scientists, and I think is rather unique. He did several fellowships. Actually, I think even before medical school, you were a fellow at the Johnson Space Center, or sometime around there, which really stuck out. I mean, I have this thing about space, and that was pretty cool. But after his neurosurgery residency, which he did at Stanford, and by the way, he was a Van Wagenen Fellow, which is a very prestigious neurosurgical fellowship. He did 3 clinical fellowships at Stanford and at Utrecht, where he did pediatric neurosurgery with a focus on epilepsy. He was recruited to the Mayo in 2019, became a full professor in 2024. Kai studies basic human physiology and the clinical translation for cybernetics, epilepsy, and functional neurosurgery. he's a real motor neuroscientist, and the way that, or movement neuroscientist, and the way that I can tell that is our common heritage. We both trace back to Sherrington. He's an extra generation than I am, but he goes through Kuffler and Eccles, I think, back to Sherrington. Yeah. So a real Sherringtonian movement neuroscientist. He is really quite interested in cortical function and how the cortex controls movement. And his lab, which is I didn't write down the name, but the goal of his lab is threefold. One is to control cybernetic prostheses. 2 is to induce brain plasticity after injury. And 3 is to study, I knew I wouldn't be able to read my writing, but distributed circuits in neuropsychiatric disease and movement dysfunction. Kai, of course, is very well published. He's well funded. He's won a number of awards, too many for me to go through now. And actually, my only other connection to Kai is that another one of my early trainees is now the medical director at his institute. That's Gelerizade, who is a neurosurgeon who is at the Mayo.
Not just the director, she's my boss.
Yeah, she's your boss. So it's we
should send her a picture.
It's a small. Well, I told her yesterday that you were coming. She had nothing but good things to say about you. I can tell you.
Could you get a photo of the two of us for Dr. Zade? She loves the photos.
Yeah, she does.
And I want to keep my job, so.
I think you will, from what she said, at least for the time being. Anyway, Kai is now going to present to us, and I can never read the titles on there, stereoencephalography as a tool for novel brain mapping and brain circuitry research.
Okay, thank you so much. So it's a really big honor to be here. And I'd like to thank Dr. Gianpiccolo for bringing me, and I would also like to thank the team here for sending Zakai here, who's here in the audience and who works closely with the group here and has done some really nice work in our lab. I won't be showing his work today just because, well, it's coming out soon, but it's really wonderful, and it does tie into some tools that we have here. So I've enjoyed my time here. This is, we got to play at Wimbledon on grass. This is Otto, who's in the top left. It's a dream to, get to come here, but we were on the fake grass at Wimbledon Park. But we did get to go visit, which was pretty fun. And then we've seen a lot of different areas around London in the past couple days with my son and I. So I don't have any relevant disclosures for this. I am lucky to be funded by a number of different institutions. I think, I think in the way that our scientific institutions in America are currently semi under assault, it's good to really note. You brought up the MSTP program. Were you part of the MSTP program as well? You're a Canadian, I guess, yeah. Yeah. So, it's good to have national institutions for training physicians, and I'm glad to be a part of that. And the NIH has been something that's been great, and it's currently being tinkered with for political reasons, which I don't think is very good. But I do think that the role that they played is truly something that we should all be grateful for, and I particularly am grateful for. My lab does a number of different things. We have on the top right, you can see some things that we basically want to build a system, kind of like what we just saw from the Dr. Brandman's group, which is, I think, quite impressive, right? So what I'm going to show you today is not as impressive as what he's shown you. What we're doing is we're working with a system that's been developed in Germany that's a 32-channel sense and stim device that we're trying to make that should be sort of scalable and robust with existing technology and would complement the kinds of things that we've seen. I'm mostly testing it in canines now, so I'm not going to talk about that device. What I'm going to talk about is our work that we do with epilepsy patients who are in the epilepsy monitoring unit. I think it's important to always think about why we get these recordings that we have. Throughout my talk, you're going to see QR codes. These point to the papers that these kinds of things come from. And there's tools for a lot of the things that you will see. There's tools that are mostly in MATLAB, and that's just because when I started, it was Fortran, and Fortran sort of evolved into MATLAB, and then a lot of people don't use MATLAB now because you have to pay for it, and I'm very sympathetic to that, but I'm also pretty old, and so it's hard to learn new stuff. So what we do is we have patients that come in for an epilepsy consultation with my neurology colleagues, and they'll wind up with if they go to surgery with one of these interventions that you see here in pink, and there's this very large roadmap that takes us from that initial consultation down to one of these therapies. And the basic thing that we start with is we ask, do the electrical changes that we measure in the epilepsy monitoring unit with EEG from the outside of the head, the brain imaging that we have in the form of MRI or other complementary techniques, agree with the symptoms that patients have around the times that they have a seizure. And so there's some times when these things agree, and those point directly to specific surgeries that we do. And then there are other times when they don't agree as well. And sometimes if we sort of know where the seizures start, but there's a border between where we think the seizure onset zone is and the functional representation that patients may have, we may do something called electrocorticography, which I'm not going to go into so much today. But in other cases, we'll do stereo EEG. And I assume many in the room are familiar with that, but for those that aren't, what stereo EEG is this process where we put these depth electrodes, each of which has about 12 to 15 electrode contacts, into structures throughout the cortex. And there are different philosophies about how this can be done in different schools, and if people have questions about it, we can talk about that after. But really what I'm going to talk to you about today are things that we do in this bedside environment where we have patients who are implanted with electrodes, and then we split the signal into a set of research amplifiers that runs in parallel with their clinical recordings, and we do a series of tasks in that context. And I'm going to show you pictures where we have interpolated activity in the brain, where we scale the size and color of electrodes. If anyone wants to reproduce that, you can get the tool at that QR code. If you like dealing with inflated brains and you want a really sort of robust system, which my little thing to make the plots that I'm showing you is not, there's this great tool that comes out of Penn, and I would just take a picture of that if you want to use it. It's from Michael Beauchamp's group. I think it's wonderful. But what we're going to do is, or what I'm going to do is I'm going to show you sets of images where we take the post-op CT, merge it into the preoperative MRI to infer where our electrodes are, and then we can plot activity there. This is what the stereo EEG electrodes look like. Thankfully, we do all of these things in metric. I don't have to tell you what they are in 16ths of an inch or anything like that. When we went from these surface electrodes to stereo EEG, there were specific clinical implications for that. So we went from something that was regular, but from the outside of the brain, to something that was irregular and sparse, but samples from deep structures, where each of these electrodes that we put in the brain is hypothesis focused, meaning we're testing the hypothesis that seizures may start in some area or may be treated by stimulation in some area. And this has enabled a number of new interventions, new to the US in terms of radiofrequency ablation. While giving a talk on this in Europe, I should always say that even though stereo EEG was sort of started in Chicago in 1949, it was developed in France. Have you seen this? I'll show you the reference. It's gorgeous. But it was really developed by Taylorac and Benko in France over the next couple decades, and then came back to the US in the 2010s. The signals that we measure with stereo EEG are very different depending upon where you're measuring from. So if we look at log power by frequency here, you can see as you go from the cortical mantle into the thalamus that your signals look very different from one another. And so we have to be aware of this when we're referencing our data. Most of the things that I'll be showing you have bipolar, so nearest neighbor reference data. But it's harder to capture things like somatotopy and retinotopy when you have these irregularly recorded from measurements. And we're sampling deep structures. And unlike electrocorticography, which was from the surface of the brain, we can really look at different nodes within circuits. And I'll show you some examples of that I think are pretty nice. First thing is to talk about some tools that we have to look at function with stereo EEG. And then I'll show you a very basic brain-computer interface, sort of silly calling it a brain-computer interface when you've seen what some of the cutting edges. This is not that, but it's still, I think, potentially useful. The one tool that I think would be useful to know mathematically is something called the Fourier transform. And really what that is when we measure some voltage as a function of time, we can translate that into what the voltage is at a particular point in time to how often the voltage reaches a particular level. And that's just saying that any signal can be decomposed into waves, where you measure power as a function of different frequencies, and that's what you're seeing here. We'll look for changes that happen in specific frequency ranges. And when those changes come and go, the superposition of all these, we call it the power spectrum. When these changes come and go in different ranges, we can then correlate that with behavior and try to use that as a mapping tool. The most basic changes that we see during behavior in stereo EEG are the following. If you have somebody stare at a blank screen, and then you measure the stereo EEG, here this is from the precentral gyrus. What you see in black is the stereo EEG voltage trace. And then what you see in pseudocolor below is something called the spectrogram, which just shows you power in color as a function of frequency on the y-axis, and time on the x-axis. And when we look at frequency, the power spectrum, as I showed you before, when we look at changes that we see during behavior, we can actually compare just resting. And when you see these peaks, these peak phenomena, here and here, sorry, my pointer working, those point to classic rhythms. So those point to things where you can look at the raw signal and you can say that looks like different waves in the signal, okay? And then we give people a cue to move their limb or something like that, and we can measure EMG in this case, and we see a change in that power spectrum. So the first thing is those oscillations tend to go away, those rhythms tend to go away that are peaked in the power spectrum. And then we have an increase in power that's basically everywhere in the whole power spectrum where you don't have those oscillations. And we capture it, we capture those broad changes across all frequencies just by looking at our signal between 65 and 115 hertz. It's not that that range is particularly important on its own, it's that that sits between 60 hertz and 120 hertz. And in the US, our power is transmitted at 60 hertz in its harmonics. I think here it's at 50. But we can capture that and those, what we call broadband spectral changes. Some people call this high gamma. I don't like using Greek letters because that implies that there's some sort of oscillation, which there's not. And we can map activity, as you see there on the right, at different locations. Okay, so this basic capturing of signal, we can talk about things, is contrasted from synchronous activity. So this would be something different, where we have stereo EEG electrode going into gray matter, and if the neurons that we're looking at and the phenomena that we're measuring are synchronized, whether that's synchronized across cells or synchronized across synapses and channels, this really means the small pieces of what we're measuring in aggregate are all doing the same thing at the same time. Okay, and so rhythm or an oscillation is a version of this, and an evoked potential, which I'll be showing you later, is also this, and that's where you have some event that triggers a voltage deflection. Of note, you may see, well, I'll tell you about it in a moment. We can also measure asynchronous activity, and this is where, if you think about how neurons actually behave, in computing populations, they're not all doing the same thing at the same time. And really the changes that we see in the power spectrum are going to look like increases and decreases in something that happens across all frequencies. And this really looks like a random walk in time. What's nice about this is that this is really a correlate of average firing rate across the population of neurons. It's been popular, so we discovered this in electrocorticography about 20 years ago, and then people tried to rename this aperiodic activity recently. So if you ever see that, that's wrong because both things on the top are aperiodic, but really this random walk or broadband process is specific for the thing that's correlated with local activity. We can generate some simple models for why we may see this. Now this is obviously an oversimplified model of how we measure the signals that we measure, but if we look at the statistics of action potentials arriving at a cell, they arrive with a random pattern that's called Poisson distributed timing, and the currents that come into a cell are effectively filtered as the charge goes from the peripheral dendrites down towards the cell body and leaks out. And if we model neurons as having basically charge separation between those areas and that as being the process that creates our voltage changes that we measure with our big electrodes, then changes in firing rate that are input to a population really give us these same types of broadband changes. And the reason why this looks a little bit different than what I showed you with data before is because it's on log power by log frequency rather than log power by linear frequency. And it's really just this average firing rate that we're trying to capture. And so I apologize for how busy this image is. This is the work of Michael Jensen, who actually will probably be applying to neurosurgery residency next year. Davis would be a great place for him to go because of his background. And what you see here on the coronal slices on the left are that if you have patients move their foot, hand, and tongue, which you see here along the different sets of coronal images, we can compare that broadband spectral change to EMG signals, and we get a map of activity where it follows the classic homunculus. And so this is a nice tool to use for functional mapping. So if we look at movement of the hand, foot, and tongue, as you see here, we can see the classic distribution in the primary motor cortex when they move these different limbs. And we can also see association areas in premotor and supplementary motor areas. And we can just map this with this simple task where we give them a cue to move and then they move. And I like using this as a tool before we do radiofrequency ablation or before we plan a resection or a future laser ablation. And this is just showing you that we can do that same motor mapping in other patients. These are coronal images, whereas the ones on the left are axial. And one of the first things I asked Michael to do before he generated those maps that you saw was just to reproduce the classic Penfield homunculus. So if you look at the picture that we've had for the last 70 or 80 years of how movement is represented on the brain surface from stimulation mapping, we knew that foot was sort of dorsal along the midline, hand was dorsal and lateral, and then face was sort of purely lateral. And I asked him just, well, why don't you just test and make sure that this representation is preserved as we put depth electrodes down into the central sulcus. And so he did that, and he came to me and said, well, I'm seeing the classic somatotopic representation, which is where these white arrows are, but at the same time, I'm also seeing areas that seem to be active no matter what part of the body you're moving or what side of the body you're moving on. And so if we look at the somatotopic delineation, now we can kind of color these electrodes based on whether they were active for hand, foot, or tongue, and you see that you have this somatotopic representation. But then there's these other areas where you have shared representation, meaning no matter what side of the body you move, or if you're talking, these things are active, and that goes down, those are down in the depths of the central sulcus. And when we look across patients and use simple clustering, you can see that they really fall deep and then they're in a few different spots. This aligned with coincident work that came out at the same time with fMRI. That's since become much more well known, I think, because they're much better at Twitter than we are. But these association areas that we call relandic motor association areas sit down in the depths of the central sulcus. We're not sure exactly what they're for, but they must have some associative role because they're not associated with movements of single limbs. And when we look at where they sit relative to the cortical surface, which is in the bottom right, that dashed red trace is basically showing you that it's down in the depths of the sulcus, and that's probably why it wasn't identified previously. Interestingly, we can use the same approach that I showed you for motor cortex and look at representation of the insula. People were not able to look very well at representation of the insula in single people before because we didn't have access to it very often. And Dr. Karazutis, if you're ever looking for a junior partner, he's coming out in about a year and a half. He's very, very good. What he did was he basically did that same movement versus rest of different limbs, so tongue, hand, and foot. And what he saw is that in individual patients, you actually have separation of different body parts in the insula. So there's a motor portion of the insula that we've known about for a long time because of deficits that people would have. And what we found is that in individual people, you actually have somatotopic separation of the different body areas. And I'm going to get back to something really cool about that in a few minutes. We can also do speech mapping. And so basically what we do for this is we have patients first do a simple noun task where they see an image and then they say a noun that's associated with it. And then we'll have them see other sets of images and say a verb that's associated with that. The idea here is to differentially recruit primary speech areas or speech production areas from speech association areas, comparing verb and noun tasks. And this approach to stereo EEG mapping can be used for other verbal tasks as well, and there's some nice work coming out of a number of groups around the world on this. So who here is an early student in neuroscience or medicine? Okay, I want you to tell me what's weird about this image. Sorry to put you on the spot like that. Just looking over here. I'll make it a little bit easier doing this.
There's a dip. Is it?
So actually, if you look at it can be a little tough on this because of radiologic versus neuroscience ways of looking at slices of the brain. This is the radiologic way, so the right part of the brain is on the left part of the image. But basically, this guy's speech representation was on the right side. So something kind of fun was it was on the right side with stereo EEG, and that correlated well with his fMRI. So it's kind of a fun thing. And so we use this for mapping. The stereo EEG-based mapping complements fMRI. So take-homes from this are just that you can use tasks, simple tasks, and looking at these evoked electrical changes to map function with stereo EEG electrodes. I think it'd be really fun to talk about this later. I'm going to show a brain-computer interface. Do you know about this article? So something that Dr. Brandman showed, a few things that I think were really nice, is he started out by saying this wasn't initially designed to be a prosthetic tool for this particular patient. When they realized how useful it was, they went to the FDA, they got approval, and that's helping that person's life every day. And I think that that's particularly neat and particularly cool. I don't know that that's representative of all the work that's out there, but I think that what they're doing in Davis is a particularly good example of how this research is done right. What I'm going to talk to you guys about are basically what we're focusing on, which is the bottom right. Can we have some sort of generalizable principles using basic brain-computer interfaces? Something Dr. Brandman spoke about earlier was that he uses a decoding approach when you have 256 electrodes. I think decoding approaches, you can really decode fine activity. What that means is you're taking the signals that you get and you're trying to have a simple mapping between what you measure to behavior, but the decoder is taking care of things. A different approach that he worked on during his PhD and alluded to is an operant conditioning approach. And that's basically where you give a patient a very simple feature. And so from our stereo EEG, we could just pick a single electrode in motor area, and we observe similar changes during imagery compared with actual movement. And then we have a very simple algorithm where we take electrode, a single electrode and that broadband power, and we map it to movement of a cursor up and down on a screen. And then we just give a patient a simple task in the epilepsy monitoring unit. Here, he's just supposed to be imagining moving his hand. This is a couple minutes into experimenting. You can see actually cheated with his right hand there. And he learns to control this cursor up and down over time. And within a few minutes, he gets almost perfect control. Now, if you have a high-dimensional signal and you have careful decoding and you're dealing with a signal that's stationary, or at least stationary for a while, a decoding approach with a large array is going to work really well. With some simpler brain-computer interfaces where you have fewer channels, an operant conditioning approach like this might be particularly useful. And what we can do is we can look at the signals that we measure, and we can compare the bottom targets and the top targets, which are in blue and red there, and the circles versus stars are indicating when it was correct trial, meaning to hit the proper target or not. And we can see how this changes over about 5 minutes. And you see that they rapidly get control over 5 minutes. And we can look across a large number of patients. I got to change this figure. The papers come out now. So it's 25 patients, and many of these are children because my practice is primarily pediatric. And if we compare overt movement, which is on the left, to imagery, what you see is that really you're able to get successful cursor control in just a few minutes if you give feedback based on mostly primary motor areas. And as you get further outside of that, it's harder and harder for patients to get control, or at least in very short time scales like we're dealing with. After 5 to 10 minutes, it's hard for them to get control. And we can do things like we can compare the map of activity that you see here on axial slices. The patient's going to get, this is just movement versus rest, like what I showed you earlier. And we give feedback based on the site in yellow in the precentral gyrus. And what we see when they perform imagery is you have selective recruitment of this dorsal premotor area. It's also near frontal eye fields. I don't know why that would be recruited during imagery, but there's some overlap between dorsal premotor areas and frontal eye fields. And I think that actually creates quite a bit of confusion in the literature when people are looking at the differences between those two things. And then we can compare that to feedback based on this feature that we've had them control. And what's interesting is I want to draw your attention to the area or the sort of inferior aspect of the superior parietal lobule, where that green arrow is on the bottom left. And you see there are areas that are only engaged during feedback. And I think that that's kind of an important thing. So with all the sophistication in the news articles and Nature papers, which we've seen some of an amazing demonstration of this today, why would you care about something so simple? And I think that if I ask, what can I do doing basic experiments with stereo EEG that helps these high-dimensional devices, it's so that we can start having some basic principles. So the first is that, well, if you want to implant high-density arrays, most of the meat of what you get, at least in these simple experiments, is going to be more likely to be in the pericentral area. And that's what we, in fact, what we saw earlier today. But this might tell you that really that's what you want to focus on rather than having arrays in other areas. Although there are people like Richard Anderson's group from Caltech who've had a lot of luck giving feedback, and specifically feedback in areas that become involved during feedback, like that site that I showed you before. The parietal reach region is back there. And that's what you're seeing here. So another principle we find is that there are areas that are uniquely engaged during feedback that aren't there during movement or imagery. And now we're doing work, I guess the audio, I don't know if this is working, but we're trying to see what happens as you enrich some of this feedback. This is just a simple one-dimensional control task, but you give them control of a basic video game. And as you have more rich visual feedback, how do visual areas and movement areas start interacting with one another? So these are the kinds of brain-computer interface experiments you can do with stereo EEG when you have, let's say, a half hour of total time to work with or a few hours total to work with. Next thing I'd like to show you is probing brain networks with electrical stimulation with stereo EEG. And we often say when we stimulate one area and measure a clear response in another area that we're measuring connectivity. It's a little bit difficult to know how to interpret that because we're giving something that's artifactual. But what we can say for sure is when we stimulate one brain area and we measure response somewhere else, we know that stimulation at one site influenced that other site. Whether that's native and normal connectivity between the two, I think is sort of a debate that's for solving far in the future, but for now, or comparing evoked activity versus stimulation. But for now, I think that we can at least say that if we're trying to figure out optimal stimulation devices, we can use single pulse electrical stimulation to do that. And so to do this, what we do is we stimulate one brain area and we measure the response of impulses that are sent from that area to other areas. And if we stimulate in cortex and we measure from cortex, people call this cortical evoked potentials. We can also do things like stimulate in thalamus and measure in cortex. And we're doing more of this. There's been a proposal lately to also call these CCPs for cerebro evoked potentials. This comes out of an institution that I used to be at where they're trying to name this. I encourage you guys not to do that because the thalamus is not part of the cerebrum. It's actually the diencephalon. So to be pedantic, we're calling this brain stimulation evoked potentials for a more generic term. Historically, this single pulse electrical stimulation approach was pioneered by Dr. Matsumoto, who continues to do a lot of really important work in this area. And these evoked potentials in stereo EEG, this shows our paradigm that you see there. We use these balanced stimulation, balanced square waves, and then we measure responses elsewhere. And we can look at about 10 responses. And when we look at the average of these are what people call cortical evoked potentials. But the responses that we get, if we look at just a few sites in different areas, have this wide variety of shapes, right? So there's no way you can look at this shape that people have used since Dr. Matsumoto's early work assumes that there's what they call the N1 and N2, or P1 and P2, and that this is a standard way of looking at evoked potentials. There's no way you can use that type of measure to describe all those shapes that you see on the left. So if there's no standard shape to think about our data in, we need some generalized approach. So what we came up with is a series of paradigms to think about these data, and then also a series of tools to handle them once we're thinking these paradigms. So the first is what we call the hypothesis pre-selected paradigm, and that's where you know basically you're stimulating and measuring from areas that should be connected to one another, right, or should not be connected to one another, as the case may be. And we call that hypothesis pre-selected because you're pre-selecting where you examine based on that. And this is work from Gabby Ojeda from Dr. Hermes' lab. She would normally be here with me. Dr. Hermes, who Zakai knows well, and who a number of other people in the room know well, is a scientist that I work with, and also my boy's mom and my wife. And what Gabby did was she looked at the effect of stimulating in hippocampus on measurements in the posterior cingulate, right, as two places in the circle of Pepys. And she then looked at the difference when you stimulate in the anterior nucleus of the thalamus. And what you see here, knowing that both of these sites are in the circle of Pepys, is you can measure relative latencies between hippocampus and ANT because you're measuring from a third site, and that gives you an internal control. So it's a nice hypothesis pre-selected internal control, and you can measure latencies between hippocampus and ANT by measuring on a third site, which I think is particularly nice. She calls this the HAP wave, the limbic HAP wave. Another thing that a lot of people will do, and I would recommend against, is basically where you stimulate from everywhere, measure from everywhere, and say, we think what matters is the voltage 100 milliseconds after you've delivered a stimulus. We call this the all-to-all comparison. I wouldn't do this. Somehow I did something badness there. Let's hope that doesn't, okay. What we can do is we can also look at what we call the divergent paradigm. And so here's where we stimulate in one spot and we measure in other places. So we may stimulate in this spot that's there in the ventral part of the precentral gyrus, measure in frontal cortex, and then measure in parietal cortex. Now it may be, the problem you can run into is it may be that the way that the place you've stimulated with is interacting with these other two sites is exactly the same, but the shape that you get is very different because the cortical microcircuitry and the laminar architecture of these brain areas are very different from one another. And so it's really hard to say that these different response shapes, if you're stimulating in one place and measuring elsewhere, really mean that the way this place you've stimulated is interacting with these other places is very different. And so that's a problem with this divergent paradigm. But we still want a general tool just to say, hey, do we have a tool that we can look at to say, we stimulate here, are there significant responses in other places? And what we don't want to do is we don't necessarily want to look at just our response here and say, oh, that looks like it's significant. We'll pick a particular time window and then look at the voltage in these responses and say that that's significant. And the reason why we don't want to do that is we're stimulating like 100 spots and we're measuring from like 100 spots. So 100 times 100 is 10,000, right? So that's too many to do by eye. We want machine learnable algorithms to do this for us. And so what we can do is we can compare the single responses like you see here. And we use this trick where we divide, we unit normalize the single trials. We divide them through by their magnitude. And then we take the unit normalized trial. So the first trial there would be in blue. And we project it into all the other trials. And we get a cloud of data that has units attached to it in microvolts times square root of seconds. It turns out that that's statistically useful because it balances the shape of the response that you have with the unit normalized trial with the amplitude of the response, and you have that from what you're projecting it into. And then we can do that not just with that first trial, but we can do this with other trials as well. So let's say we took the, the 10th trial there, and we projected it into others. We get a different cloud of data. And if this total cloud of data that we get from projecting all trials into one another is significantly different from zero, and you can see that all of these are very, very different from zero, right? The axes don't even start until 70 in these units, then we have a significant response. And our algorithm learns this without us having to know anything about it, which can be particularly useful. And then we can, we actually will throw away half, so each pair of trials is only counted once. You only normalize it. So if I took trial, let's see, 1 into 10 here, like I've shown you, and 10 into 1, we actually throw away the ones that are mapped into each other. You only take half of them. So if you don't do that, your statistics can be unbalanced, and you can get false significance. And so if this cloud of data is different from 0, we know we have a significant response. And what I showed you was for throwing a second of data into the algorithm. But let's say I've thrown a second of data in here. That doesn't make a lot of sense because it looks like the signal drops away after about 100 milliseconds. So what you're seeing on the right of this plot in the middle here is sending in a full second of data, and that big black dot is the average of all of those projections. And unlike the last slide, you can see some of these projections are less than 0, right? So it still looks like it's probably significant, but there's no reason why we should have picked a full second of data. We could take a half a second of data, or we could take 100 milliseconds or even less, okay? And we have the average of those projections, and we can take the profile of the average of those, and that's that dark black line, and we can plot that over our average response. And that tells us the moment at which our signal starts dissolving back into noise. So now what we have is we have a machine learnable technique to discover how long an evoked potential is significant for. And it seemed to me that people should have been doing this 20, 30 years ago, and we had the computational power to look at the classic EEG signals and so forth, or even for neuromonitoring, but they haven't. And so this is a machine learnable tool. If you guys want to use it, the QR code is here. And you can see how this example that I've shown you in this slide differs from the previous slide. And armed with this, we can now talk about our voltage on a single trial K here as a function of that, this is a single trial in black, of a shape that we call C of t in red. And that shape is then scaled by just a scalar alpha, okay? And then you have a residual noise that's there. And so because you have, are you asking me a question?
Yeah, can I join my math class in the hallway?
Yeah, sorry. Double timing his dad. So on single trials here, you have signal and noise, and you can talk about signal and noise for single trials of stimulation. So if you're doing neuromonitoring or something else, this gives you that. And you know immediately when you have a non-significant response, like the bottom, or if you have a significant response of different types, like in the middle and the top. And you can compare, even though these shapes here are wildly different from one another, you can compare what the voltage magnitude of the response is. So this is like RMS voltage, but only the RMS voltage that corresponds to that shape on a single trial. And you can also look at signal to noise, which would be 4.5 for the top one and 1.6 for the bottom, or explain variance, which is in the right column. And so this turns out to be a nice generic tool for looking at interactions between sites. Coming back to what we found out about insula, we can look at this site that was a significant site during hand movement in the insula, as you see there, and compare it to a site that was significant in hand motor cortex of M1. And we stimulate in hand motor cortex of M1 and measure responses, and we see they're significant in insular hand area. When we look at stimulating hand area of M1 and looking at insular tongue area, it's not significant. And then we can do the converse. We can stimulate primary motor tongue, and we don't get a significant response in insular hand area, but we do in insular tongue area. So this expands upon what we found before, where we have somatotopic representation in insula. And not only do we have somatotopic representation, but the somatotopic representation is somatotopically connected. So hand area of M1 and hand area of insula are connected to one another in ways that they're not cross-connected. And then we can do the same looking at, this is looking at tongue area of the right and left insula to one another. The slide's a little complicated. I'm sorry if you're interested in it. This just came out, and you can look at it there. But it shows that you have reciprocal connectivity between somatotopic regions in insula. The reason why the stimulation can be useful as epilepsy doctors for stimulation is we can basically use this to test stimulation strategies when we use multi-electrode stimulation. So if we identify areas that are that have epileptic spikes, we can then ask ourselves, well, does test stimulation in these sites change seizure frequency? And if we stimulate in the anterior nucleus of the thalamus, we see that interictal spikes are reduced in the limbic circuitry on the left, but not nearly or not as statistically robustly in occipital cortex. But then if we stimulate in pulvinar nucleus, we get very significant reduction of interictal epileptiform discharges in occipital cortex, but we get no changes in these epileptic events in limbic circuitry. Only before we decide to implant pulvinar, let's say for occipital neocortical seizure suppression, we may want to know if the part of the pulvinar that we're thinking about is actually connected to the cortex that we want to suppress. And so we can use a single pulse approach to do this. So what we're going to do is we're going to stimulate at that site in green and that site in yellow. And one is in the medial pulvinar, the other is in the lateral pulvinar. And basically, when you stimulate medial pulvinar, you get significant responses in temporal neocortex, but not in the occipital lobe. Conversely, lateral pulvinar lateral pulvinar electrically influences occipital cortex. So this says that you can't just stimulate pulvinar for posterior quadrant epilepsies, you need to be stimulating the proper part of it. And we can use stereo EEG to establish this. And so some examples where we use tandem targeting for connected circuits would be things like centromedian nucleus combined with SMA and pre and post central gyrus. And so now moving away from what I've shown you, you have a generic tool for hypothesis pre-selected and divergent paradigms. We can look at a convergent paradigm as well. That is where you measure from one place and you do single pulse electrical stimulation in many other places. And here, when you get different shape responses, because you're always measuring from that same patch of brain, different shaped responses means that you have fundamentally different types of interaction because you're always measuring from the same patch of cortex. So if that input is coming in the same way from stimulating at both areas, it should have the same shape. And if it's not, it should have different shapes. So this allows us to assess whether or not there are different motifs for influencing the area that we're measuring from by stimulating at other areas. And here, because we're using this convergent paradigm, different shapes really do imply different connection types, which was not the case in that divergent paradigm. And just to illustrate this, this is now electrocorticography with a patient I was doing lesional resection for. And we're measuring here from the parahippocampal gyrus, which you see on the bottom left. And we can deliver electrical stimulation in the superior temporal gyrus, measure a response here in the parahippocampal gyrus. We can then stimulate in the fusiform gyrus, and you see you get a very different shaped response, right? So that tells you that the shape of the voltage response that you get in one patch of cortex is very much dependent upon where you've stimulated. And so that tells you the way that that area that you're measuring from is influenced by stimulating at other areas is fundamentally different. And what I'm doing here is this is that same example, but now we're plotting on the surface of the brain. And I'm not plotting responses that you get in those areas of the brain. We're always measuring from that parahippocampal site. We're plotting at the place that you've stimulated the responses that you get in the parahippocampal site. And when you look at these responses, what you see is that there's these clusters that emerge visually. And what we'd like would be a way to identify these algorithmically. And so I took these responses that we had, and I thought I could do some kind of standard hierarchical clustering approach. And it turns out there's nothing that really fits statistically for that. And so what I did is I called for help. So this guy, Klaus Müller, runs a machine learning center in Berlin, and he's been sort of a friend and mentor of mine for a long time. And he helped me develop this clustering algorithm that is generally applicable to brief time series data like I'm showing you, and specifically applicable to our electrical stimulation. And I'm not going to go too much into it other than to say that we calculate significance matrices using that same single trial projection technique like I showed you before, only now we're comparing across different types of responses. And then we reduce the significance matrix that we get from that using something called non-negative matrix factorization. And what that does is that performs clustering for us for free. So that we see, hey, there are 3 basic shapes that we measure at that place that we're measuring from based upon where you've stimulated. And we can map these 3 different shapes back to the place that you stimulated with a magnitude of response that we elicited by stimulating at those places with the same stimulation paradigm. And so what we saw visually, we now get for free algorithmically. So the take-home points from this are that you can use this convergent paradigm to get interpretable motifs and connectivity to tell you if there are different fundamental responses you get in one spot depending upon other places that you've stimulated. And basically, it gets us this, what we call basis profile curves that we can then map back onto our original sites. And so I'd like to take these tools that I showed you and for about 5 minutes show you how they can be useful for single patients to discover new therapies for people who have relatively rare conditions. And I'm going to show you a patient who had post-anoxic myoclonus. It's called Lance Adams syndrome. I'm going to show you some videos of this. He gave consent to show these videos. Basically, he had anoxic brain injury after an infection. And let's hope this works here. Can you guys hear the audio? And so this is Dr. Clausen, my movement disorder colleague that I work most closely with. And you can see this patient, when he tries to engage in standing or engage his limbs, he has an extreme difficulty doing this. Okay, so this is him just trying to perform basic tasks. Now, what we did is we did stereo EEG. I'm not going to read through the full ethics review that we did before we did this, but I do think it's important if you're going to start doing things that are non-traditional or non-standard to involve an internal ethics panel early. I think that beyond just being sort of an intelligent thing to do to make sure that your institution believes in you, I think it's also gives you some peace of mind. And what we came up with was to implant stereo EEG into a number, and with the American insurance system, this can be quite difficult, particularly many of these patients are disabled and they don't have private insurance, they have government insurance. And he had seizures about once a month, general seizures, and that was sufficient for them to say we could do this. And we did this stereo EEG implant, so targeting globus pallidus interna and externa, VIM, VOP, subthalamic nucleus, and then also centromedian nucleus of the thalamus because of the epilepsy. And we wanted to know what's the right place to stimulate. So and we also implanted primary motor and primary sensory areas. And I got to write this up at some point soon. But basically, this was our construct. And what we did is we implanted him and then monitored him in the epilepsy monitoring unit. And we first had a baseline period, and then we went through stimulating for 4 to 5 hours at a time, and then did clinical testing to see if he was getting helped. And then we also did some tasks. So I'm going to show you some changes from primary motor cortex, like you see here. And the first thing we can do is I showed you before the stereo EEG responses. Now it says healthy subject. This is a patient who has epilepsy, a different patient was implanted. So they're not healthy, but they have a different disorder. And then our patient, without stimulation, you can see he still does have these standard electrophysiological changes, but there's something odd going on with the oscillations that he has. They don't seem to go away quite right in the way that they do in most other individuals that we measure from during behavior. And I'm jumping to the punchline here to show you centromedian stimulation, but basically we were able to change the oscillation center frequency that he had during movement and rest, and that seemed to give him dramatically improved function. I have no idea why. And then we stimulated all these other sites and didn't have quite as robust changes. This looks like a Jackson Pollock painting here, so I'm sorry about that. But what I'm showing you is along the x-axis are all the different sites of the brain that we measured from. And we're seeing this measure of activity during movement and rest, and the different colors of dots tell you where it was being stimulated when we were doing this. In pink, you see that that jumps up immediately, and that's in the motor cortex specifically, comparing movement and rest, and that was centromedian stimulation. So if we look at his ability to, or our ability to generate these functional maps, comparing movement and rest, you see in primary motor cortex, there's some activation comparing movement and rest, but it's not as nearly as robust as when we have centromedian stimulation. So that told us there's something special about centromedian stimulation compared with stimulating anywhere else. And then we could look at these evoked potentials like I showed you. And we start out first by applying that CRP technique, or that first technique for parameterization to figure out what the longest interval of time we should use is. And then we looked at clustering, the basis profile curve clustering technique, to see whether or not there were unique channels in the primary motor cortex. And what we found is that there were three basic types of changes that would be induced for stimulation in primary motor cortex. The first was stimulating other areas of primary motor cortex. That's the green voltage deflection that you see there. The second, and this is the output of that clustering algorithm, so we didn't look at any of this by eye or anything like that. So this is just what the machine told us, right? The machine algorithm told us this. The second was the classic sites we would associate with basal ganglia, subthalamic nucleus, and not just what we call VOA and VOP, ventralis oralis, the basal ganglia inputs to thalamus, but also VIM nucleus of the thalamus. Now, it turns out that those areas do have some crosstalk between one another. So when I talk to patients, I say VIM nucleus of the thalamus, that's where you get cerebellar inputs. That's the classic story. But then I talk to people like Peter Strick, who mapped all these things out, and he tells me the story is much more nuanced, and he knows a lot more than I do, so I trust him. But the cool thing here was that centromedian nucleus alone in no other area produced this unique shape in the voltage response that we see there in orange. So that tells us that there's a unique stimulation pathway between centromedian nucleus and primary motor cortex that's different from stimulating other sites in motor cortex, and also different from stimulating other classic subcortical targets. And then when we look at our clinical responses here to stimulating different areas, this is him trying to speak. And since I'm running out of time, I'm going to skip to CM here. So he had very significant improvement selectively during centromedian nucleus for speech. And then this is comparing just baseline function to centromedian stimulation. Now, the areas that people had stimulated before in the literature, and there's only a few case reports of this, had been globus pallidus, pars interna, or VIM nucleus of the thalamus. And so what you can see, particularly if you look at him trying to do the sort of chin to finger to chin, or run his leg up and down, is that centromedian nucleus was and you can see it with the ball task too if you wait long enough. Centromedian nucleus was dramatically better than baseline, and it was dramatically better than the other sites as well. And my neurology colleagues naively, let's see if I can get to the next slide, naively scored this, and what we saw was that centromedian was by far the best, and second and third best, or second best, sort of tied across the board, was VIM with GPI and GPE. We could use this Boston Scientific 4-lead device that has these 8 contact linear arrays that we can get both GPI and GPE at the same time as we stimulated centromedian nucleus with a directional electrode. So CM was directional, the other one was longitudinal. So this has produced significant improvement for him. It's not great, but it's better than he is without it. It also produced improvement in his seizures. So that brings me to the end of my talk. What I wanted to talk to you guys about today is how we can use stereo EEG as a tool for functional mapping to look at different brain circuits. We can do some basic BCI experiments. Obviously, these are going to have to be very basic, very episodic. And then how we can use stereo EEG to come up with novel therapies in one particular example. It's really an honor to be here at the birthplace of neurology and neurosurgery. And I lastly wanted to end on a plug. Maybe sometime we could bring people to Rochester to interview you guys here. I'd really like that if we could interview you about your patient. This is a podcast that we have. The guy on the far left is an Oxford professor now. I know some folks here work with him, named Tim Dennison. So he was one of our first guests. And I'm part of a very large team. I'm very grateful to get to be part of that team. Thank you guys so much. Sorry for taking so long.
Excellent talk. Thank you, Kai. Questions?
Thank you so much. My name is Francois. For context, I'm a neurosurgeon resident with an engineering background. I'm doing neuro AI conditioning. And so my question is about state dependence in neuromodulation. So the question is, if I intervene in drug-resistant epilepsy, a patient, and the short-term clinical and neurophysiological improvements are present, how do I tell whether this is genuine network reorganization, like SOZ, easy, change to late?
I'm really sorry. Were you able to hear better?
You can't hear me?
I can't, I'm sorry.
Okay. Can you hear me now?
Yes.
I'm Francois, neurosurgery resident.
Nice to meet you.
And neuro AI PhD currently. And the question's about understanding the mechanism of a clinical effect. So, a DRE patient, you try a stimulation strategy, you see short-term clinical improvement and biomarker improvement. How do you know it's not suppression versus real network reorganization versus you fix sleep fragmentation? How do you know it's one or the other? What kind of signals or experimental design allow you to trust your thinking behind what's actually happening?
Okay, so the short answer is that you don't. So what he asked, for those who couldn't hear, is or could everybody hear? My hearing is not great. So basically, if you think about what we do with stimulation, it's not very well studied what we're doing when we do it, but you want to know, are you interrupting an area? Are you driving an area? Or are you changing the dynamics in that area? Those are sort of the three short-term things you can do. And then longer term, you might ask, do you change the general dynamic state and the ability to go in and out of dynamical states that a particular area has, or the way that that area interacts with other areas over time? Teasing those things out, I think, will require more subtle experiments that we do in the epilepsy monitoring unit. Epilepsy monitoring unit is a great place to study different phenomena in brain circuitry. I do think that for people who have therapies that are not great, and there are a lot of not great therapies out there, we should be treating other diseases the same way we treat epilepsy, right? So one great example is dystonia, right? So dystonia right now, if somebody has a genetic dystonia, they respond very well. If they have a secondary dystonia, they do not respond as well. And I think it's because there are a constellation of things that have dystonic presentations, and some patients will benefit and some don't. And maybe we should be testing them first. As far as, let's say, suppressing an area, well, 130 hertz in many cases is probably simply, which is our standard thing is 130 hertz, but I know it's different in different centers. It's generally probably suppressing an area, but not always. And when not always, I think there are probably more clever people than me in the room to answer why and when that is. Lower frequency stimulation, I think, can drive, and particularly can drive specific circuits if they're on the same time scale as that lower frequency. So if you're delivering 5 hertz stimulation, if you have a circuit that's resonant at 200 milliseconds, there you go. Something that I wonder is that if you think you have a disease of brain dynamics like epilepsy, why do we give regular stimulation at all? And so I think more and more we need to be moving to a regular timing in stimulation. We have a finding that we're now cleared. I guess it's the publication is in submission, and there's a patent pending on it showing that noise things that obey the statistics of particular kinds of noise are particularly good at still interrupting pathological phenomena, but without producing side effects in quite the same way. But I think that the first thing when we attack any problem, just like I showed you for electrical stimulation, you want to do is you want to just organize your thoughts. And for me, the organization of the thoughts are we interrupting? Are we driving? Are we short term? or are we disrupting dynamics or changing dynamics? Okay, and then in the long term, how are we changing the circuits? And that can be something that's purely local. That can be selecting for particular types of interactions over other types of interactions, and those can be driven by stimulating at different timescales. But I don't have it figured out beyond that. I just think you have to start out with some very clear criteria to organize your thoughts, and that doesn't mean that those criteria are going to be the correct set of criteria to look at. It's just a starting framework. And then if you look at most of the things we have in medicine, it started out as a particular framework that was subsequently refined and improved, but you just start by organizing your thoughts, and then only later do you try to articulate what your analyses are going to be or what your sets of paradigms you're going to use for stimulation are going to be from that. So I didn't answer your question at all, but that's how I would start.
You did. Thank you.
Hey, I just wanted to ask, in terms of like picking sites for like electrical positioning, both as like stimulation for if you're looking at connectivity or for therapeutics, and also even in terms of like its like use in sort of like the movements. You mentioned kind of moving away from the M1 seems to sort of make it less significant of a signal that is correlated to movement. What about a consideration of something like the cerebellum, which obviously has like strong correlations to movement?
For brain-computer interfaces?
Yeah, exactly.
So, I mean, I think the cerebellum can always, when you start speculating, you can always put your foot in your mouth. And again, when I make some comment like this, there's usually somebody in the room who knows a lot better than I do. But when I think about the types of disorders that we see of the cerebellum, many of those disorders are related to feedback processes that optimize and tune the dynamics that we have for brain activity, right? So that, the classic experiments that you do. But for brain-computer interfaces, the feedback and tuning that you get is going to be mostly visual. And while there are presumably inputs from visual areas and corticobasal ganglial thalamic areas that we normally think of projecting into cerebellum, I think that's going to be a lower yield place to go than primary motor. Some other questions I would ask that might be useful along the same lines would be, that I would have for Dr. Brandman, would be things like, if you want to drive M1, maybe you need to be stimulating thalamus to stop the rhythmic entrainment that goes on, that we measure in these oscillations, at the same time as you're trying to decode M1, so you don't deal with the problem that your signals are inherently non-stationary because the brain states are inherently non-stationary. Maybe you can make them a little more stationary by disrupting corticobasal gangliothalamic feedback loops, which we're used to doing because that's what we do with DBS all the time. So that would be one thing. And then the other thing I would say is you asked about the cerebellum and what it means for brain-computer interfacing. I would ask, if you make somebody decerebrate and their function, they still have some ability to function, they're very, 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 very discoordinated. So what does a BCI based purely on cortex look like without that cerebellar feedback? And, really that's a much better question for people who work with patients that are implanted long-term for brain-computer interfacing, which, frankly, I've done a lot with electrocorticography and stereo EEG in the epilepsy monitoring unit, but I don't have a lot of longitudinal experience, and as of yet, I haven't entered into that. Not because, basically because I think it's a really difficult problem, and I tend to be very opportunistic in my research. I work with patients trying to take advantage of clinical situations that we're already in to learn. But I think trying to understand those processes that will take longer periods of time, you need people like we've already heard from today, but maybe they should be doing, something like stereo EEG monitoring at the same time as they put in their arrays, and then they pull out the stereo EEG electrodes. I don't know, but see how you change the dynamics that you care about for feedback. And that's a real science of brain-computer interfacing. One of the biggest criticisms I think I have of brain-computer interfacing as a field is that it's, for the most part, a large series of tech demonstrations rather than a set of general principles that have been learned for brain-computer interfacing over time, rather than being something where people say they try to learn about motor cortex or motor circuitry and then translate that into BCI, or language and language circuitry and translate that into BCI, but there's less principles related to brain-computer interfacing. It's mostly just really cool demonstrations for one case at a time. Now, the history of neurology and neuroscience is one patient at a time, right? HM, right? Broca, what's his name? Phineas Gage, right? These are like the classic ones, right? And probably that's in this building. So I don't want to make too much media about myself. So I would say those are the things I would think about along the lines of what you're asking. And I think that the stereo EEG environment can be good for learning some of those principles. The stuff that I'm doing where I have chronic implants and canines, mostly we're just looking at signal stability of the device. We have some really cool canine functional representation stuff that was incidentally, but that was very incidental.
David?
Yeah.
I wonder if you could tell us more about thalamic ECG. So it's something that is not common. So as you might know, in the UK we cannot do DBS for epilepsy, and we do not normally do thalamic ECG. And it's very debated whether thalamic ECG would something that would be advised, because in this case we cannot do DBS, but also has been long said that it's not possible to predict the effect of DBS on the base of thalamic ECG. But I think you were showing some data that actually this might indeed be the case, and I was wondering if you can tell us more about it.
So I guess we had this paper that just came out. There was a series of 4 papers. Now, so there's a friend of mine, Dario Englot, I'm sure you know Dario well. I was talking to him about how we were going to have this come out because they had 4 opinion papers, and he said people stopped thinking about this 5 years ago, like it's not controversial anymore. What I can tell you is that there are centers who do thalamic stereo EEG, and they're doing ANT, CM, and pulvinar in every patient bilaterally. That's probably going too far. But the truth is that in our practice, and really we're primarily looking at CM, ANT, and pulvinar, and you guys could, there's 3 other articles. I think actually, if I'm honest, I think ours is really good, and I think Mark Richardson's is really good. But that's, little vain maybe, but hey, I'm a neurosurgeon, So what we use thalamic stereo EEG for, I do it based on the hypothesis for circuits that may be involved, is for the following. First, seizure timing, right? If you think you have a seizure onset zone and that's kicking in after you picked up stuff in the thalamus, you don't have your seizure onset zone. The second is the test therapeutic stimulation, like I showed you before. That's the biggest one, okay? Because for most of our patients, we do test therapeutic stimulation of cortex and of thalamus, and I'm putting those electrodes in to know whether or not the patient will at least have a reduction in, and if they have a high enough seizure rate, then you can just look at reduction in seizures, but at least in interictal discharges, I want to know, is a stimulation strategy I might use for this patient going to be viable? And I don't care how good your center is, there's no way that your patients who make it to stereo EEG and are extratemporal, no, I don't think anywhere has better than 50% seizure freedom. And I would say most places have far less than that, and I think ours has far less than that. And so given that that's the case, you always want to have stimulation as a backup. Even if it's not your initial therapy after stereo EEG, it's your therapy after that. So I think it's worth having thalamic stereo EEG. And in the US, we also have some weird stuff, like we get rejected a lot for, I'm fighting insurance companies all the time. And I can say to them, look, and you should not implant somebody to fight insurance, that's wrong. But I can say, we know their seizure activity is decreased when we stimulate in this area where I'm proposing to put this stimulator in permanently. Yeah.
So the follow-up question is that how much your seizure control rate has increased since you started to do this? Because so of course you have some information about the fact that the seizure might decrease during stimulation, but do you have evidence that the seizure rate, so your seizure burden has decreased in DBS? Because so at the moment, for example, we only have VNS. We discussed before what we think about VNS, but if you can prove that actually in this technique DBS is so much better than VNS because you have a way to target in a way that gives over 50, 60, 70%. That's huge, and we would also use it to leverage to start to do thalamic ECG and DBS.
Well, I think we know that at least for ANT, thalamic stimulation improves And the other thing actually is when you are fighting insurance companies, I never say where I'm stimulating, I just say thalamic. Thalamic DBS, sorry, has been shown to reduce, because the nuance is lost on the urologist that you're talking, or the rheumatologist you're talking to about where you want to put your device. The ability to do this and get good results depend upon if they have a high interictal epileptiform discharge or a high seizure rate. Long term, it's really hard to say. I mean, what we do know is, what I can tell you is I stimulate, sometimes I put in 4-lead constructs, that Boston device. And we may have had, let's say, initially equipoise where we didn't know the ANT or CM was going to be better. We thought CM was going to be better based on stereo EEG, but we said we should do both because we don't know. And then CM turned out to be better, right? So we did what we would have done naively, and then we found that stimulation in the site we thought would be better was in fact better later because we were stimulating both, because we really wanted to hedge because we could. But saying overall that that doing thalamic stereo EEG produces more seizure improvement in DBS down the road than not having it, I think will require a large multi-center trial just to get the numbers, which I think would be cool.
Last quick question to Beata.
Yeah, it just, I can't help but ask.
Thank you so much for making it, by the way. I know that.
Yeah, I just have to ask about the spikes. I'm sorry about that. To ask a neurosurgeon about the spikes, because that's what you're using often as a readout, isn't it? It's whether you find a connection and whether you think that the network is indeed driven by a particular thalamic nucleus that you're sampling from to predict whether DBS in a particular nucleus might be better than another. And B, what you showed in your trial there was basically the potential of modulating during the SEEG
Yes.
the spikes.
Yes.
Now, for an epileptologist and someone who looks at these SEEGs, we do know that spikes are a very poor predictor of whether a seizure is about to start. So how good do you think is this going to be a marker, a biomarker for your subsequent success?
So the first thing I should say is that when it comes to interpreting spikes, I'm probably not better than someone you pull off the street. But my neurologists are, I think, quite good at that, and I do trust them. The second thing I would say is that for most patients that my colleagues show me, the site of where seizures are initiated often have more interictal epileptiform discharges than other sites that we measure from in the same patients. And if you see a reduction in interictal spikes, that doesn't mean that every set of interictal spikes you see is the one that causes the clinically significant seizure. And so it may be that there are some sets of interictal epileptiform discharges that are suppressed and others that are not. And I think that's when we have our most nuanced conversations. And most of the time I listen there rather than trying to, I mean, the neurologists I work with know a lot more about that than I do. I know that's a cop-out, but it's why we're a team. But what I will do is I will, in the cases where we have, let's say, people saying, I think based on the spikes you should stimulate one area or another area, I'm always pushing to do both with a 4-lead stimulator. So I don't, the other thing that we have access to in the US is we have access to this responsive neurostimulation device, the NeuroPace device. And that in some cases has allowed us to look at least what we would infer to be spikes over the long term, and the correlation between spikes over the long term and detected spikes, however you interpret that from the NeuroPace device, and I don't know how they interpret that. They can look at the relative frequency of those over long-term stimulation, and my understanding is that the improvements in clinical seizures are correlated with reduction in interictal spikes in that device. That's my understanding, though, and you may have more understanding already about that than I do. So I know that's a cop-out, I'm sorry, but I have to trust them.
Perfect. Perfect. On that note, I think we'll end. There is a reception outside, and I'm sure that both David and Kai, I'm not sure, but I hope that they'll both be out there for a bit so you can ask more questions if you have more questions. But
we'll be out there.
Okay, perfect. So Kai, thank you very much. That was a tremendous talk.